a quick sort of check for people. So that was a great fun question, but now I want to try to get at least 50 people to answer for this data set here, this Swiss roll that we've been looking at. We were talking about intrinsic or latent dimensionality. Those terms are sort of the same thing. How many latent dimensions are there in this data set? Just a quick quiz, pop quiz to see what people are getting. Awesome, great. So we got enough responses here. Very, very informative, great. So let's see. So the correct answer here, I think there are two correct answers. It could be 1 or 2, but this is not the overwhelming response, which is 3. And so
well, if you add 1 and 2.
Yeah, if you add them together, that's correct. If you're 1 or 2, gives you 3. But so this data set, the Swiss roll, just to clarify what these terms mean, I think this is always really important to do these kind of quizzes because sometimes this happens, and I want to make sure that everyone understands the difference between intrinsic and ambient dimensionality, which has come up a few times in the group in the chats, and we wanted to make sure we address that now. So the ambient dimensionality of the data is 3. And I can understand the dimensions that you're measuring are 3, x, y, and z here, but the latent dimensionality is different than the ambient dimensionality. So we've been talking a lot about how for the Swiss roll, you have to walk along the Swiss roll to calculate, to sort of stay on the data set within the data. You cannot, there's some regions of these 3 dimensions that are not valid combinations for this data set. And so you could imagine there's basically, I think, if you count 1 latent dimension, it's actually the distance from the center of the roll. So it's sort of this axis here where we're spinning around the outside. There's that dimensionality. There's also, you could interpret the width of the strip. So this is like a piece of paper that's been rolled up on top of itself. And so there's the axis along the edge here, but there's also the width that we have. There's some width to the data set. And so you can imagine that that width is also a potential latent dimension. So the correct answer here is 1 or 2. And it's always important to think intrinsic or latent dimensionality will almost always be less than the number of ambient or measured dimensions of the data.
Can you actually go, maybe, okay, this slide has it. Okay.
Yeah.
That's where.
Would you like, do you want to clarify, Smitha?
Oh, no, I just wanted to go off of it and into what we're gonna discuss first here. So one thing that's interesting about this Swiss roll data set is that, as Dan was mentioning, you could think of it as having either 1 or 2 dimensions. If you think about 1 dimension, it's the dimension of the coil. The second dimension could be important, but it actually looks like the second dimension is some sort of noise, right? Because the spread isn't very high in that dimension. It just looks like, there's the main trend into the data is this, and there's just a little bit of variability due to measurement or something along this line. So in this data set, it stands to reason that there are actually 2 intrinsic dimensions, but one of those dimensions is more of a noise dimension. And if you were trying to denoise the data, which is the first topic that we're going to talk about today, you might want to get rid of the second dimension and sort of project everything on that first latent dimension that you have. So there's this very powerful idea that has been in signal processing and other image processing that your eigenvectors, the eigenvectors that describe your data space, whether it's PCA eigenvectors or eigenvectors of your affinity matrices, the more prominent longer range eigenvectors are signal, whereas the eigenvectors that sort of explain less variability could be noise. And that's actually kind of what I'm going to be talking to you about, because we can apply the same kind of idea to denoising single-cell RNA sequencing data. What about the coiling part? Can you clarify that question, Ziba? Or you can ask it in Slack and we can talk to you in more length about it.
Sure.
Oops, I don't know, it's not advancing. Okay, got it. Okay, oops. Now it like advanced a lot. Okay, so so we talked about the idea that data has an intrinsic manifold, and these intrinsic manifold dimensions. I'm sorry, my kids are home. Guys, you can't scream. I'm in a lecture today. Please, no screaming. Okay, thank you. They have homeschool every Wednesday. I don't know why. All the other days they go to regular school. So the idea is that you have this low dimensional manifold, and some of the dimensionality reduction methods that we actually talked about yesterday actually help you learn the intrinsic sort of coiled dimensions of the data. And as I mentioned, that these dimensions of the data can be useful for denoising the data. And so we're going to talk about these same kinds of concepts that we talked about yesterday. Affinity matrices, Markov normalized affinity matrices, their eigenvectors. And this is going to be very useful for denoising. It can also be useful for batch normalization and batch correction we're going to talk about, and also for compositional analysis, which is reasoning about how different single-cell data sets are composed differently and how that reflects on the underlying experimental condition. So first I'm going to talk about denoising data using a manifold model. To motivate this example, I'm going to start with something really simple. So this is the MIT logo if you look at it carefully, but it has a lot of noise on it. It's sort of like salt and pepper noise covering this whole thing. You can think of the number of dimensions in your data that are sort of independent as the rank of the matrix. And you can think of the noise as additional dimensions. So you might notice that if you think about this image as data, you can think of the rows and columns as the features of the data. Then it's not broken up that way. Each kind of pixel in this image has both noise and signal, which is the main image you're looking at. But it's possible using dimensionality reduction to change that and to have your noise dimensions separated from your signal dimensions. And then what you can do is you can simply, once your dimensions are broken up in such a way that your noise dimensions are separated from your signal dimensions, you can kind of recreate the data without the noise. So how do we do this? How do we get dimensions that correspond to signal and noise? Think about some of the things we did yesterday. Any suggestions? Are the responses being shown here, Dan?
For some reason, it doesn't look like Poll Everywhere has grabbed the slide yet.
Okay, but some people are talking to me in chat, and there are,
I'm just going to try to make sure it's it's open. Okay, it is.
It's okay. Because it looks different than when it grabs it, right? So some of the answers I'm getting on chat are PCA, and somebody further specified that by saying eliminating the last PC dimensions. So that would work. That's a really valid way of denoising data. It might not be the best way of denoising data. And one of the ideas is because PCA, as you recall, breaks up the dimensions so that the highest amount of variation is in the first PC component. The second highest amount of variation is in the second most. So the last PC, which some of you mentioned in the chat, explain very little of the overall variance of the data and are likely to correspond to the noise. So if you reconfigured your dimensions so that they're PC dimensions rather than the originally measured dimensions, you've kind of altered the balance so that you have the signal variation or the main variation you're interested in the first few dimensions, and then the last few being sort of noise. You can also do this without PCA with nonlinear dimensionality reduction methods, but I'll sort of come to that in a second after I talk about this. Someone else said SVD. So the interesting thing you can realize is you can find the PCs, which are these eigenvectors, also using SVD. It turns out to be mathematically equivalent. But usually, technically, you are using SVD. So we'll talk about that in a second. I'm again having, okay, there we go. Okay, so SVD stands for singular value decomposition. It's similar to eigendecomposition for non-square matrices. So when you do SVD, you're actually doing an eigendecomposition of this matrix that's M times M transpose. But it turns out if your M is your data matrix and it's mean-centered, then M star is actually your covariance matrix. And so PCA actually gives you the components as well as the loading, or SVD gives you the components as well as the loadings. But you can use SVD to kind of throw out the noise dimensions, because what you have here are values that explain how much variance is in your data in these components. And so what you can do basically is you can eliminate some number of eigenvectors and corresponding eigenvalues on all three of the SVD matrices you're getting, and then you'll get a matrix that's the same dimensions as before, back in your original dimensionality, because what SVD does is rotate, stretch, and rotate, just like eigendecomposition does. So what you've done is you've taken your data, okay, you've rotated it into these eigendimensions, you've dropped some of the eigendimensions, you stretch in the appropriate directions, and then you're zeroing out some of the dimensions that you don't like, and you're rotating back. So now you have a matrix that's back in your original dimension, but it doesn't contain these noise components. So the basic idea is you're recreating your matrix without those eigenvectors that you feel correspond to the noise. And of course, there's a parameter hidden here too. So if you low rank approximate this MIT matrix, you see it's very clean. It just throws off all the noise because this noise is sort of independent or linearly added. This is from this Medium post. The main reason that this works is because I would like to say that the main signal that you're seeing is sort of smooth. And so it's sort of what we would call a low frequency trend. Whereas noise can be jumpy, can change very quickly. It can change very quickly from pixel to pixel, or dimension to dimension, or it can change very quickly from observation to observation if you're looking at a graph setting. And so by taking off things that change quickly or are high frequencied, you can restore the smoothness of the signal, which corresponds to the main signal. This is the idea of denoising. But there's also always this question, how many eigenvectors should we use to reconstruct the data? And how do we know which of them are noise? Any thoughts on that? I think this one is active.
Yeah, it looks like this one's working.
Yeah. Our friend the elbow plot. Like they hear friends with elbow plots. Okay, yeah, so that's where can one find the recording from yesterday's lecture? I'm sure Dan can send that.
That is a great question to ask in the MISC help question channel on Slack, and I will answer you there.
Okay, scree plot and judgment. Okay, these are all good answers, and there is no exact answer to that, absolutely. In machine learning, you always have this question of model selection, which is subjective. There's no machine learning algorithms that have completely eliminated human intervention in terms of model selection. I don't know what a jackstraw is actually, but it could be. There's always things I don't know. So the magnitude of the eigenvalues is exactly what you would be looking at. So this is an example of a scree plot where you're looking at the magnitude of these eigenvalues. And the idea is that the amount of variance explained by signal dimensions is different than the amount of variance explained in noise directions. So noise can shift things off in random directions, and there might not be a long trend there in that noise dimension. And so in your scree plot, you can see what are called eigengaps. So often, as a heuristic, people choose a location of an eigengap to cut off their data, and they would eliminate, for example, the eigencomponents that correspond to these eigenvalues down here because they're not explaining very much variance. And so the idea of a scree plot is that you are visualizing the magnitude of your eigenvalues, and in PCA, the magnitude of your eigenvalues is the amount of variance explained. And so you can just eliminate those. So single cell RNA sequencing data is also extremely noisy. I would say it's even noisier than a lot of the image data that we've been showing you because of a phenomenon called dropout. Incidentally, you have to be careful when you use the word dropout in machine learning because it means a lot of other things to people working in neural networks particularly. The idea is that if you have a cell, you more or less reached into this cell and pulled out a random set of transcripts, and the rest of them are dropped out randomly because the capture efficiency is not that high, and those beads have captured some random number, people estimate between 5 and 10% of your RNA molecules. So this adds, heavy duty noise to single cell RNA sequencing data. The question is, can we use ideas from image denoising to denoise single cell RNA sequencing data? For example, the PCA method that I just showed you. And most of the answers seem to be yes, with caveats. I would love to hear those caveats. So the answer is yes and maybe. Yes, because for sure that method works and you can eliminate noise. But what you have to remember is that in PCA, the new axes you're finding are simply linear combinations of the old axes. And if your data is sort of rotated from the original axis and where it lies, so there's certain genes that are covariant and certain others that aren't, and that's it, then you might be able to use PCA directly. But as it turns out, low rank approximation using SVD, which is related to PCA, is not the best option if you have noise that goes along the manifold of your data, particularly if the manifold of your data is nonlinear or curvy in the original data space, which most of the data sets in single-cell RNA sequencing tend to be. So here is an example. Let's say that you have this data set which is rotated into high dimensions. And now let's sprinkle some Gaussian noise, but let's do it along the data. So this is kind of like you're mostly getting the right measurements, but they're like splayed off from where they're supposed to be because of noise. So this is spread out. So low rank approximation can't do anything because it's trying to take off planes in your original data space. It's not going to be able to catch this coiled dimension and get rid of it. Whereas a method that I'll talk to you about called MAGIC, which uses nonlinear methods and the affinity matrix, can, because it learns the intrinsic path of the data, and then you can get rid of it from that. When you have Gaussian noise plus something like dropout, it's even worse, because the dropout noise is pretty severe, that you won't be able to correct it all unless you use nonlinear methods. So what nonlinear methods are we going to use? You guessed it. Instead of PCA, we basically use something like a diffusion map. But it turns out we actually don't have to eigendecompose again, and I'm going to go through how we do it. So the idea is, again, that you have a cell by gene matrix that you turn into a cell by cell distance matrix, as we did in the last two lectures. We convert those distances to affinities by pushing them through this kind of a Gaussian kernel. Again, we use an adaptive bandwidth because that recovers the geometry of the data better during diffusion. We Markov normalize it. Once you Markov normalize it, you get what's called a diffusion operator. You can take the eigenvectors and the eigenvalues of that diffusion operator, and using these, you can do what we did in PCA, more or less, which is you can try to take off the last few eigendimensions and retain the first few. But there is actually a really sharp analogy between these and what's called low-pass filtering or frequency manipulation in signal processing that allows you to do it in a slightly different way. So if you remember what these eigenvectors were, the eigenvectors of the diffusion map as well as the Laplacian eigenmap that we saw yesterday, these eigenvectors actually directly correspond to frequency harmonics of your data. So remember that the second eigenvector, which is corresponds to a low frequency trend, and your last eigenvector corresponds to a high frequency trend. So what you can do is you can get rid of exactly this kind of high frequency noise by having any gene or any value that's defined throughout your graph, which is all of your gene measurements. You can just eliminate their high frequency components directly. So just to give you a picture of this, you have your cells, they're all connected by edges if they're close by, because you have the cell-cell affinity graph. Now any gene you can think of as a signal on this cell, and you can take this signal and you can decompose it into those eigenvalues and those different eigenvector components, which actually gives you a Fourier transform of sorts. And then you can just eliminate the high frequency components and take the inverse Fourier transform. And this is this is how you can directly use even the signal processing analogy we had in this setting. And this is from the field of graph signal processing. So the eigenvectors of your Laplacian, also your diffusion eigenvectors, are the frequency components, And to load your data to them, you take your signal and you just multiply it with, you just take your signal here and you multiply it by the transpose of your eigenvectors to take these inner products. These inner products will tell you how much does this signal load on the lowest frequency eigenvector, the middle frequency eigenvector, and high frequency eigenvectors. So now you have a frequency decomposition of whatever gene or signal, which is a column in your original data. So for this signal here, it has some low frequency components and has some high frequency components. So now what you can do is you can actually artificially eliminate these high frequency components, zero them out. And then you simply take the inverse Fourier transform, which is very easy. It's just multiplying your graph Fourier transformed signal by the eigenvectors without the transpose. And then you get it back into what's called the vertex domain. And now you cleaned your signal. And you can do this to each and every one of your genes. The question about what's low and what's high frequency on a graph is a little bit tricky though, because a graph doesn't have a regular structure. So in this graph, if this was the connectivity, this signal would be quite low frequency because these genes are, whatever the value of this gene is slowly changing. On this end of the graph, it's high, and this end of the graph, it's low. But if your affinity matrix changed, the frequency spectrum of the signal would change. So this is a translation of what happens in very regular structured domains like time and space onto the setting of an affinity graph. And that's what the graph Fourier transform is all about. So once again, you have a question of how many eigenvectors should you take off? So how many eigenvectors do you low-pass filter when you do this graph Fourier transform?
Also, just while we're doing this, would like to mention for people who maybe have not heard the phrase Fourier transform before, there's a bunch of links to materials on our workshop page under graph spectral theory that I think will be really helpful if you have like some sort of foundational questions about these topics.
So if you take off too many eigenvectors, you're going to be left with very little information. If you take off too little, it's going to be still quite noisy, and you might miss trends in your data. And your matrix may seem like it has a lot of missing values. So there are several sort of heuristics around to figure it out. But actually, one of the solutions that we came up with, we don't directly decide how many eigenvectors to sort of take off. What we do instead is we softly filter the eigenvectors. So some eigenvectors are left mostly alone, and other eigenvectors are downweighted rather than taking them off. And this is called low-pass filtering rather than low-rank approximation, because we're not entirely taking off eigenvectors, but we're applying a soft filter to the eigenvectors. And this is our method MAGIC, which was published like almost 3 years ago now, or 2.5 years ago now. This is what it does. So the way it achieves a low-pass filter is we actually power the Markov affinity matrix. That inherently creates a matrix where you have eigenvalues weighted by the eigenvectors, but the eigenvectors that are close to zero have gone down in magnitude more than the eigenvectors that are prominent. And now we simply load every gene back to this powered Markov matrix. And what that means is you're recreating the genes with the high frequency eigenvectors damped. What this looks like in the vertex domain is, in the vertex domain, what you're actually doing is you're recalculating every gene as a weighted average of the value of that gene on your graph neighbors. So close neighbors get high weighting, distant non-neighbors get low weighting. You have this weighted average that's replacing everything. So if you're trying to recalculate the value of the gene of some gene on this cell, you'll get high weighting from your near neighbors. You'll get lower and lower and lower weighting from these things that are farther away. So what you're doing is this kind of smoothing, and it's a graded smoothing. And so low pass filtering in the spatial domain or the graph domain corresponds to smoothing. What you're doing in the frequency domain, and so there's an animation here that shows as you increase the T, you get a more and more smoothing and less high frequency components. So if you're increasing the T with which you do it with, you're getting more and more low pass filtering and a smoother signal on your graph. But in the frequency domain, what's happening is you're eliminating eigenvectors from your signal. And the higher you low pass it, the fewer of those eigencomponents are being left alone. And so if you, with no smoothing, here's a Fourier transform of a gene. If you use the magic soft filter with T equals 2, you get this profile. If you use T equals 5, you get this profile. T equals 100 gives you a very severe profile where you've probably oversmoothed. And that's what it looks like in the frequency domain. So what does this actually do to your data? This actually, to a large extent, restores values that you could think of as missing, but they're just dropped out. So we make no distinction about which value is missing and which value is not. Basically, every value is sort of undercounted or dropped out, but if you apply magic with different T's, you get recovery of these signals, and the recovery of these signals can facilitate the learning of gene-gene relationships. And the recovery of the signal you get is non-parametric in the sense that it can take on any trend or shape throughout your data, because what you're banking on is the fact that without the noise, cells that are in similar parts of the data affinity graph have similar gene expression profiles. Now, if there are genes that don't follow that, and most genes seem to, then those genes you couldn't recover. If the gene value could change very rapidly from cell to cell, or if it's a very temporal thing that's constantly fluctuating. But the main things like the phenotype of the cell, usually the cell cycle also, most of the trends in your data you can recover by this kind of denoising. Okay, so the question of what T should we choose? The main idea of choosing the T is the same logic as choosing how many eigenvectors that you want to select, but in the low-pass filtering case, we choose it a T. So what one heuristic that we proposed in the paper was this idea that the values stop changing after a while. If you kind of if you can imagine the values diffusing from cell to cell, they stop exchanging information. So T of 6 is going to give you results that are very similar to T of 7, and when it stabilizes like that, that's the T we would use. There was a more recent paper that said that that technique tends to oversmooth, and they tried a different technique called molecular cross-validation. What they do there is they take their mRNA data and they actually add sort of ambient noise or dropout artificially by reforming artificial cells, and they look at which T recovers it back to the ground truth cells that they had. And they tend to choose pretty small T's, like 2 or something like that. And so there's different heuristics possible, and this is an active, ongoing area of research. So how did we validate a method like this? So we used data that actually didn't have dropout, and we induced dropout on it, but the data had manifold structure like your cells do. So these were worms that were developing over time, and so the worms at different developmental times, these worm embryos are related to each other by this kind of simple manifold structure. and they're very small, so they have small sample sizes like single-cell RNA sequencing. We eliminated data, we dropped it out. We did it in a variety of ways. One of the ways is to set a bunch of values to 0. And then you look at certain trends like time trends or gene trends and see if they're recovered by magic, and it's recovered quite well. Not only that, because magic isn't making specific assumptions about dropout noise or anything, there's other kinds of noise that are cleaned up too. So you actually see that the imputed matrix is a lot cleaner than even the original matrix before dropout, because we also eliminated Gaussian noise and other kinds of measurement noise and things like that. So this is the idea of basically manifold denoising, manifold-based denoising of single-cell RNA sequencing data, which can be helpful. Somebody talked a little bit about missing value imputation versus denoising. So I do want to make a distinction here. The denoising methods that I've talked about that are strongly rooted in signal processing and image denoising assumes that the data has some noise. So every entry could be corrupted by some kind of noise. On the other hand, there were people earlier on especially who tried missing value completion methods, or what are traditionally called imputation methods. These methods require you usually to know which values are present and which values are missing. We didn't necessarily feel that this was the situation for single-cell RNA sequencing data. It's not that you know that some gene value is missing. It might just have been dropped out. Compare that to something like a hospital setting where you know somebody didn't take a test and you're trying to impute what the value of the test result would have been, or based on their other results. So missing matrix completion and other imputation techniques are ways of filling in missing values. And there is some literature in the single cell world trying to incorporate these methods, but generally it's very hard because you don't know what true zeros and false zeros are, and there's been some controversy and people trying to say, oh, this isn't a true zero, or is this zero inflated, like are there just a bunch of zeros added where people zeroed out values. But usually the noise assumption, which is milder and seems more realistic, does seem to be easier to work with in the single cell setting, where, as I said, you can kind of think of it as you grabbed a bunch of transcripts randomly from your cell and left a whole bunch of others out. Okay, so just to go back, we have compared magic to low rank approximation, which I talked about at the beginning of this lecture. It turns out that low rank approximation can get sort of the linear part of the trend correct, but oftentimes you're not getting the nuances and the nonlinear shapes correctly with those. So it's not able to correct the noise in these very loopy nonlinear parts of the data, which are abundant. And this is real data. This is real data from a human breast cancer induced EMT system. These are real genes from that system that we're showing how we recovered it. This is the matrix completion method, which is more meant for missing data and doesn't work at all. So this is just an experiment to show the difference between using denoising assumption, linear, and nonlinear. So so I've presented denoising, but it's actually really important to consider what downstream tasks actually need this denoising. And when you should use it, because this is a pre-processing method that is going to alter your data. So when should you be using it? And so just thoughts on when you should use this additional pre-processing step. Okay, so I'm getting a bunch of answers here. Okay, and I would say that the two that look the most correct to me are trajectory analysis, well, three now, trajectory analysis, gene trend analysis, and differential expression. It turns out that you don't really need denoising for visualization because there are visualization methods like Fate that already denoise. But if you want to use PCA, you're right. You'd have to denoise your data first with something like Magic, and then if you did PCA, it would look a lot better. But you could use something like Fate or diffusion maps that already does the denoising internally. Clustering, it turns out that you actually probably don't need the denoising for clustering because clustering is anyway clumping a bunch of cells together based on overall similarity. And it turns out that you have so many dimensions that you can pretty successfully guess on the overall similarity. Because remember, in order to do the denoising, we had to learn a cell affinity matrix. And that affinity matrix is reasonable, otherwise we couldn't even do this denoising. And so by that same principle, you could use that same graph that you used and just use like Louvain or something, which is a graph form of clustering. So the clustering method can have that built in, and you can decide which are close. So just to go over 2 examples of tasks where I would recommend using denoising. Oh, this thing keeps going really fast or really slow. Okay, so one application that I would say is, I'm really having trouble with this, Dan. Can you go to the first slide there? Okay.
Which slide do you want? This one?
Yeah.
Okay.
So one application that I would recommend using it is if you have some clusters or sets of cells and you want to identify what they are. And somebody did mention cell type identification, so thank you. That was correct too. What type of cell they are. Often, some of the really characteristic markers, like transcription factors and surface markers, are the very ones that are very afflicted by noise or because they're lowly expressed. Okay, so if you restore your genes and correct it in this way, you can get a lot more of your surface markers and transcription factors, and you can see which part of the cellular manifold they're expressed in and whether your cluster lies in that part or not. So it's very useful for restoring characteristic markers and identifying cell subtypes. So GFAP is a marker for astrocytes. Without magic, you have no idea where the astrocytes are, and now suddenly this cluster lights up and you're like, oh, there are the astrocytes. So this is one example of where you would use it. The second one is in understanding gene-gene relationships. Because if you only take 5% of the transcripts, let's say you have some gene value that's counted in some of the cells and it's dropped out in other cells, then you might never have 2 cells that have both gene values, both for the gene in the x-axis and the y-axis, by random chance. But the diffusion and smoothing of this value will make it so that you see the trend and you can recover quite nonlinear and multiple trends which could occur in different clusters using this kind of method. If you use it too much, you could start coming up with spurious correlations. So those are some situations where I would recommend using data denoising. The summary of this part is that data denoising is a diffusing or smoothing of values over a graph. It has strong roots in low rank approximation, but it's a linear, soft kind of low rank approximation. More diffusion or more steps of diffusion, for example, in Magic, is more denoising. If you have any questions, please submit it on Slack. So with that, I'm going to give the control over to Dan so he can talk about it.
Wonderful, thank you. Okay, so just again, direct everyone to continue the conversation on Slack. And I would like to, right now, I'm going to talk about 2 different things. So just maybe to clarify a little bit, maybe we should have done this at the beginning about the syllabus. So I know before the last time we did this workshop, we talked about clustering today, but we're actually switching and taking the last part of this lecture is going to talk about analyzing multiple single cell data sets, because you'll see, I think, about how some of these ideas about denoising and smoothing and using graphs are somehow conceptually related to how you might actually quantify the effect of experimental perturbations. And then we're going to do some clustering later. So the next topic I want to talk about is batch correction. This is another sort of way that you might be altering the data in order to better interpret or draw biological conclusions from it. And so batch correction sort of has a lot of different kinds of definitions, and people mean different things, and I talk about phrases like batch effect. And so I want to maybe clarify how we define batch effect in this course, and understand that you might find slightly different definitions in different papers or from different groups, just in the same way dropout has different kinds of meanings. So batch effects in single cell data, we think about as systematic, non-biological differences between samples due to measurement conditions. And so there's a lot of different reasons why that might happen, depending on exactly what measurement technology you're using. So it could just be things like that, the actual ability for you to create cDNA libraries might be biased based off of, the temperature or humidity of the sample preparation process. Maybe there's some problem with the way that those cells are being captured in droplets in a 10x machine. If you're doing mass cytometry, there's almost surely going to be problems with calibration or antibodies that could, or maybe not almost surely going to see that, but if you have batch effects, that's a very likely culprit. Has something to do with quality of the beads. I think it is also sometimes used to refer to true biological differences between samples, but those that are uninteresting to the particular hypothesis or biological questions under consideration. And so I just want to, those two things are a little bit different, but at the underlying fundamental property here is that there's some difference in between samples of a data set that you'd like to not consider or remove. And when how do you identify a batch effect? So often what will happen is you'll do some sort of visualization of the data, perhaps using PCA, you could be using FATE or UMAP, and the plot that you get might look like this. So here we have cells from two different donors, and the maroon cells are from donor 1 and the purple cells are from donor 2. And when you create this embedding, even though these may be from peripheral blood cells, we know that we should find the same sorts of cells, natural killer cells, T cells, B cells, eosinophils, et cetera. When you actually plot them, you don't find, for example, that T cells are overlapped with T cells and the B cells are overlapped with B cells. You find that they're very different across the data, across that visualization. And so this kind of analysis, this kind of inspection of the data, I think, is one of the first ways that people often identify a batch effect in their data set. There are some quantitative measures that you could use to actually say whether or not there is a substantial difference in between the data that you're observing across your samples. You could use a measure of diversity or measure of entropy across your samples to assess the degree of mixing. I think one of my favorite ways that this has been done in the single cell literature is a method called KBET, which actually batch effect test, the K nearest neighbor batch effect test is what it stands for. And the idea is to take each cell. So here, this is a bunch of cells that are sort of normalized and dimensionality reduced. This is not necessarily a visualization on like UMAP or FATE, but just maybe on your 100 PCs. And here we're looking at, essentially 3 different samples here as marked by the color of the cells. And you look in neighborhoods, K nearest neighbor neighborhoods, and say, how many of those neighbors are from the same sample versus different samples? There's not necessarily a right amount. If you have 0, so for example, in this data set, the neighbors of a lot of these purple cells, there's going to be 0 neighbors from a different sample. Same thing for the maroon. None of the neighbors of these cells are from that purple sample. So if you have no mixing, then that's absolutely a problem. It's going to make it really hard to do a lot of the downstream analysis that we might want to do. But if it's greater than that, there's not necessarily a perfect number. You could do comparing to references, but the goal of batch effect correction is actually to improve these measures of mixing between data sets. And so if you were to apply a batch effect correction tool to that PBMC data set that we were looking at before, the result should be to realign those 2 data sets to actually get a good degree of overlap between those samples. So one of the things, I mentioned before, I was showing that on fate, you can see very clearly that there was separation, but you don't necessarily need to use fate. A lot of times, people like to just sometimes use things like UMAP. They're just going to go straight from loading their data to generating a UMAP or fate embedding or a t-SNE embedding. But oftentimes you can actually identify these kinds of batch effects just by looking at PCA. And so here I'm showing you, we're actually looking at the first 5 principal components of this data set. This plot is PC1 versus PC2. This plot is 2 versus 3, 3 versus 4, and etc. What you can see is that in some of these, I would say in one, there's definitely a huge difference between these two samples. There's also somewhat of a difference in PC2, but there's a little bit less in PCs 3 and 4 and PC5. And so there's actually more mixing there. And so the first question that I want to ask you is, looking at those principal components, I'd love to know if you guys have any ideas of how we could actually use that information and, sort of thinking about how we described PCA yesterday, how could we use that information to actually remove the batch effect from the sample just using PCA without using any more sophisticated algorithms? I'm going to wait for some responses to come through before we move forward. Are people okay, here we go. So drop low value eigenvalues, select those PCAs that separate by patient, regress out the PCA from the matrix. Yes, so I think all of these answers, or remove the first PC, all of these I think are very good ideas. This is exactly what we're sort of hoping to hear, is that you could either just remove PC1 and only take PC2 or greater. So it'd be one way just to literally drop it and then only build your graph from PC2 and above. You also, when you do principal components analysis, you get a loading of the genes and the principal component. And so you could actually, if you didn't want to just look at your dimensionality reduced representation, you could actually remove genes or reweight genes based off of the inverse of those loadings. So decrease values of genes with higher loadings on PC1, or even just remove those genes entirely from your data set if they end up being things like ribosomal proteins that you're maybe not really interested in investigating. So absolutely, these I think are all really good ideas for how we could use PCA. And so in this data set, if you were to just drop the first principal component of the data set and then train fate on the rest of it, you can see that there's actually pretty good, batch effect correction that you're seeing in this data set. It's not as good as what I was showing you before, but there's actually, better overlap within some of these populations. At least now when we're doing a visualization using fate, we're seeing some overlap. And if you were to measure things with the K-bet statistic, it would have improved. Sorry, Smitha, did you want to say something? Maybe I just misheard. I heard an artifact in my speakers. Great. Okay, so PCA, right? This is one way to do it, but it's not perfect. It's not exactly what we'd like to see. And so there's some limitations here. As we've mentioned, PCA is a linear combination of genes. And so if you have a non-linear batch effect, some genes, go up or down with different respect to others in a non-linear fashion, just like we were showing with magic, that there are some non-linear patterns in the data that we might want to preserve. It's not necessarily going to be possible to just do batch effect correction by removing principal components. So there is one approach to doing this is using CCA. And so CCA stands for canonical correlation analysis. And if you are familiar with some of the data integration methods as part of the Seurat package, there's actually a tool called Seurat integration, which really fundamentally is just CCA been rebranded to be part of this software toolkit. But fundamentally, the idea here is called CCA. And the idea behind canonical correlation analysis is that you're using the matrix, sorry, let me just get this right. So you're using the matrix of cells from condition A and condition B. And what you want to do is learn a new embedding of the data set. So you're not just doing PCA on one data set and then PCA on a second data set, or even just concatenating them together. But the goal here is to find linear axes of variation across the two data sets. So a common set of variation across the data sets. And so this is also a linear combination of them. Imagine instead of just taking the covariance across two different genes within one data set, you're actually looking at covariance across data sets and trying to find axes that maximize those common variation across the data sets. What you can do is derive a set of linear axes through both data sets that gives you some kind of matched basis, some new basis for both of them. And so CCA is actually quite similar to PCA, but the main difference here is that instead of just concatenating the matrices, combining all of the cells from both samples and then doing PCA, we're actually understanding that there are differences between them and trying to identify what are the axes that are common to both of them and not weighting as heavily variation that's unique to one of them. And the unique component is what we would consider the batch effect. And so then by matching cells along these axes of variation, we can actually align the two data sets that your visualization here shown using TSNE works a little bit better. So this is another linear method for identifying common axes of variation in the data set. But one of the problems is that it assumes to some extent that you have across almost all of the data sets some shared axes of variation. And so if there's one cell that's present in a data set or cell type that's present in one data set and not present in another, CCA can get thrown off by this and think, well, that's a unique kind of variation. I don't want to preserve that when I'm learning this common basis. And so another way that one could think about doing this kind of batch correction is instead of trying to jointly learn an axis of common variation from both of the data sets using the covariance across them, you could think about using a tool like harmonic alignment, which is a method that we've developed in the lab. Sorry, I'm realizing that on my screen some of this text is a little bit tiny, but in the slides you can zoom in as much as you want. You imagine having two different data sets in which there's some kind of batch effect between them here. We then learn a graph representation of each of the data sets together, and then look at the spectrum of the graph Laplacian. Just like we were talking about the eigenvectors that we're using for magic, you can imagine taking an eigen decomposition of that graph, getting the vectors of different frequencies over that graph, and then trying actually to match, maybe diffusion component 1 actually aligns with diffusion component 2 of the second data set. If, say, for example, there's a rotation and a stretch, you could imagine that PC1 actually better, it's not actually principal components, but that the diffusion component 1 of the graph better aligns with a different diffusion component, but they just need to be matched properly. And we show that if you do this when we say band limited, we mean we don't want to think that diffusion component 1 matches up with diffusion component 200, because those are much higher frequency. We want to match low frequencies to low frequencies, mid frequencies to mid frequencies, and high frequencies to high frequencies. We can then actually realign these spectral representations of the graph, go back from the realigned version of the data to the original data set, and realign the data in the ambient space, and then create a representation where actually there is better mixing between them. And one of the nice things about that is now we're looking at diffusion components of a graph, which are now nonlinear. So we have this ability to take in not necessarily just linear combinations of variation across data sets, but actually nonlinear variation across data sets, in the same way that we show that magic works better than low rank approximation. And so one of the things that we found, however, is that this kind of rotation does not necessarily match all of the cases that you might be looking for. I think this is the last method that we're going to cover today, is a tool called mutual nearest neighbors. Yes, and this is the last one. So I've talked a lot about this idea. What if there are some cells that are present in one data set but are not present in the other, and there are still some correspondences between them? So the idea behind mutual nearest neighbors is actually quite similar to the idea of k-nearest neighbors that we were talking about before. So in this algorithm, the goal is actually just to keep the ambient features the same, but to learn a graph representation of the data in which there is no batch effect, in which there's good mixing between the data sets. So the question is, like, how do we just build a graph that in that process removes a batch effect between them and respects the fact that there may be some cells that are present in one data set and not in the other? The way that this is done is by not just linking k nearest neighbors, but by looking for all the cells joining k nearest neighbors. So in this plot here, all of these, this orange cell here is connected to this blue cell and this blue cell. This blue cell is connected to these other two orange cells. And in this second blue population, this top cell is connected to this orange cell, but note that that orange cell is actually not mutual. That connection is not mutual. Some of these are mutual. Some of this orange and blue connection is mutual, but some of these connections are not. And so the idea is actually to only connect cells that both have each other as mutual nearest neighbors across the data set. So these two cells are mutual nearest across data sets, but this purple one is not mutual. And so then when you do the alignment, you can actually create a set of vectors in the ambient space or just build a graph representation of the data where you connect here, say, these red cells to these red cells, these blue cells to these blue cells, but these pink and yellow cells, which have no mutual neighbors in the other data set, have their differences preserved. I think this is an important way to understand how you can build graphs here. One of the things I want to note now too is that we can actually use mutual nearest neighbors with magic to do denoising. I was showing before that you can find some set of vectors that correct the data sets together. And what I want to point out is that when you do this, you're creating a graph, and this is the adjacency matrix for this. This is an adjacency matrix representation of the graph. This graph has a block structure. So on the block diagonals, you have connections. So imagine that these rows correspond to cells in sample 1. These rows correspond to cells from sample 2. And similarly, the columns are cells from sample 1, and the columns are cells from sample 2. And the values in this matrix are the strength of the connection from whatever the cell is on the left to the cell on the right. So this square are just the connections within sample 1. This is a set of connections within sample 2. And then on the off diagonal blocks, you have the cross sample connections. So this is, you said these are the rows for sample 1, and these are the columns for sample 2. So these connections are from 1 to 2. And so if we were to use this kind of affinity matrix, what we can do is do smoothing over this graph, this cross data set graph, to actually pull the data together across samples by strengthening what connections that may not otherwise have existed from 1 to 2. So these off diagonals are actually the key connections that we're going to use to do magic across these kinds of data sets. Showing you a comparison of these, there are a few other kinds of batch correction methods that are not exactly what I was talking about today, lemma and combat, and uncorrected data. And lemma and combat are both linear batch correction methods that work somewhat similarly to the way I was talking about doing correction with PCA or CCA. But what I want to emphasize here is that we have 3 different kinds of cells denoted by color, and then cell types denoted by batch. Batches denoted by shape. So we have some mixing here within cell type 2, but cell type 1 and cell type 3 are not mixed. There is a batch effect between them. And one of the things that's really nice about mutual nearest neighbors is that this actually correctly aligns cells of the same type and doesn't create any spurious connections across types. Whereas some of these linear methods either fail to identify a correction vector between cells that we know should be mixed, or also create some spurious connections between cell types that we know should be distinct. And when we do this kind of correction, combining a method like mutual nearest neighbors with magic, one of the things that we can find is that actually this correction gives us a representation of the data that we can then use to create another dimensionality reduced version of the data where there's better mixing between the data sets. And so I want to just ask a question now. So is, just to make sure everyone sort of is following and understands what we're doing, is mutual nearest, oh, I should have hidden these responses here. Is mutual nearest neighbors an example of linear or non-linear relationships between the data? And I want you to think about why it might be linear or non-linear. So I'm going to wait until we have, let's see, we got 130 people on the call, so I'm going to wait until we have 50 responses here. I want to get a good sample. Seven more people, come on, we can do this. Awesome. Great, okay, wonderful. So 90% of you said nonlinear, which is correct. And the reason that mutual nearest neighbors is nonlinear is because it is a graph-based method. And graphs are built using kernel functions, which have nonlinear relationships. So there's nonlinearity introduced by the kernel that then says that it's not, just a linear function of distance that creates the connection between them. There's a nonlinear function of distance, but then even more so, we're upweighting connections or only considering connections where there is a joint relationship where cells are mutually connected to each other, and then downweighting connections where only one cell is connected to another cell in a data set. To summarize this section, I know this is a very complicated in detail topic that we're sort of just going through in about 20 minutes or so, but I want to sort of wrap up a little bit of this conversation of data denoising. So we talked a little bit about how batch effects are sample-specific changes in the measurements that you have across samples of a biological system. The goal of batch normalization is to align cells of the same type across data sets. So that you align cells that are both found across samples, but that you do not force connections between cells that might be isolated to only one sample. And that one of, I think, the most popular batch effect correction methods, this mutual nearest neighbor method, normalizes batches by matching cells that are both close to each other and not cells that are far from one sample to a second cell. And so I really encourage you, I know that this is a very complicated topic and you probably have a lot of questions. There's a ton of methods out there. I really would encourage you to post questions on Slack, and our TAs and Smitha can help answer any questions you might have about how they work. In the last 20 minutes of the lecture, I want to start talking about something else. But before we leave batch effect correction, I actually want to take a little bit of a step back in time and talk about, I think, the optimal way to reduce batch effects in your data, which is proper experimental design. So when you have your data and there are batch effects in them, once you measure your samples, load them up, and you're sort of looking at your data in Python, to some extent, it's a little bit too late. You can absolutely salvage data sets this way. But I think that it's really important to think back before you do the experiments, to think about some of the principles of experimental design that are going to give you the best opportunity to identify or test the hypotheses that you have in your experiment. And so the first thing, I think is really important to do is to have randomization. And so randomization was first defined as like splitting your population randomly into treatment and control groups. But really the goal of this is to try to get your treatment and control populations, your multiple samples, to be as homogeneous as possible. And so in single cell, randomization really is trying to talk about how do we make sure that there are not systematic biases in the populations of cells that we're sequencing in the treatment versus the control sample. So you want to make sure that they're the same genotype, for example. If you can process samples in the same day, run them in the same machine on the same chip. There are even some technologies like cell hashing, where you can actually tag cells from different samples with antibodies and then mix them together and split them across lanes so that you don't even have biases in the 10x machine or in your dropseq of cells in the treatment or experimental condition getting handled differently. And I think it's really important to think about all of the different ways that you can ensure homogeneity across your experimental and your treatment and control samples. Another thing we're actually going to talk about a little bit right now is the importance of replication. If you can, it is absolutely better to do your experiment more than once. Don't just have one treatment and one control, but actually do it multiple times. I'm going to show you why that's so important. And we'll do that in the coding notebooks today. And then finally, everyone likes to talk about controls. Think about what exactly are the negative controls that you want to have. Make sure that this is all set up properly. And I think by thinking about some of this ahead of time, hopefully you can reduce the batch effects that you might find in your data set. Great. So finally, I'm going to spend the last 15 minutes today talking about compositional analysis. Before we get too into this, I just wanted to let everyone know that I gave a lecture on this topic last week at a single cell seminar series at Yale. It is available online for free. You can go to bit.ly/meltlecture. This is a 1-hour lecture on just this topic. And I am cutting down all of the content from that lecture into 15 minutes today. I expect that there will be a lot of questions about exactly how this algorithm works, but please, I really encourage you to go check out this lecture, and I'll share the link on Slack for a much more in-depth, detailed explanation of how the algorithm works. So we talked at the very beginning about how single cell data sets, we have measurements across tens of thousands to millions of cells, tens of thousands of genes, and now we're starting to get multiple samples. And we just talked about how you might think about integrating those samples. So what if you have the data, it's integrated fine, and now what you want to do is ask the question that you've probably been really trying to get to this whole time, which is, what is the difference between these two conditions? So the classic experimental setup here, let's imagine we're just going to have two samples. You have one control and one treatment sample. You add some, experimental condition to the treatment sample. You generate single cell profiles from each of them. You get out two different data sets, a set of cells and genes, and you know for each cell which condition it came from. The way that we want to approach tackling this kind of problem is to think a little bit back about this whole idea of cells being observations sampled from a manifold. So we were talking before about this underlying cell state space, a latent space of the data, and that there's some underlying density over that state space that in a given organism, tissue type, et cetera, it's going to there's going to be a density that's going to dictate the frequency with which you will observe cells of a given type. So this might be an example of one of those density functions. So imagine that these are just two axes of cell state. They could be T cell, this could be naive to exhausted or regulatory to effector, but there's going to be some density associated with this kind of cell state. So these two might be relatively high. In the back here, we have some cells that are going to be a little bit less frequently observed. And in these black regions, you might expect not to see any cells at all. Like maybe those are neurons and you're looking at blood cells. And so there's going to be no data density there. So this underlying function, this is our model of what's going on, that we're drawing observations from this function in our control condition. And then what we do if we have a treatment is that we're somehow altering that underlying density function, that probability function that dictates the frequency with which we'll see different kinds of cells. So some kind of cells might be more likely to be observed. We're going to see more of them. They'll be enriched. Some cells are going to be less likely to be observed. And then some cells might be totally unaffected. We expect to see exactly the same number across data sets. And so really, in this context, using this sort of manifold idea, what we would like to do to quantify the effect of the perturbation is compare these two density functions and look at the difference between them. Identify what are the regions that are enriched after your perturbation, what are the regions that are depleted after your perturbation, and what are the regions that are unchanging across conditions. And what's important to note here is that this is quantifying the experiment as an effect on an enrichment or a change in one condition relative to another condition. We're not trying actually to quantify this underlying data density. What we want to do is compare the two of them and see where one is higher and the other is lower. In the full lecture, I actually sort of compare this to another paradigm you might use to do this kind of comparison, but I'm actually just going to today talk exactly about how we've decided to tackle this using an algorithm called MELD. And the way that we try to quantify that density is that we take the cells from both conditions, we concatenate the data matrices, and we build a cell similarity graph. So we might be using a Gaussian kernel function to build that. In one of the data sets in the manuscript for this paper, actually, we consider a data set of pancreatic islets from human donors, where there are some batch effects across donors. Those batch effects we know were driven by expression of genes like insulin and glucagon, classic hormones for pancreatic islet cells. And so we apply a mutual nearest neighbors graph across donors, but not across the experimental condition. So you could use there are many kinds of graph construction strategies you might use that might include some of the data correction approaches we talked about earlier. It's important to note if you apply correction across the treatment and control axis, you might be removing an effect of your perturbation. Once we have this cell similarity graph, we think back again to this idea of signals over the graph. So one of the signals that we're really interested in is a signal that represents like an indicator for each cell, whether or not that cell came from a given sample. So here we have two signals, one corresponding to the treatment condition. So this signal is going to have value 1 for cells that come from the treatment condition and 0 elsewhere. And then there will be a second signal. There's going to be one of these per graph. This is a second signal that's 1 for all the cells in the control condition and 0 elsewhere. So as we mentioned before, these are graph signals. They're vectors that have a value associated with each node in a graph. And what we want to do is estimate the density of those signals over the graph. So if you noticed before, I was talking about trying to quantify a change in density over an underlying ambient state space. So we don't actually want to necessarily do this across the full ambient space in a continuous setting like you might do with a classic kernel density estimate. But instead, what we do is extend the continuous, or we consider a discrete representation of the kernel density estimator over a graph to first quantify the density of each of the samples over the graph. And then, sorry, I thought there was another slide in here, excuse me for that. But what we do is we take those densities for the experimental condition, do it again for the control condition, and then we calculate the ratio between the two of them. And then using that ratio, we can then for every single cell in the data set, obtain a measure of how enriched or how much more likely we would be to observe that cell state in the treatment condition relative to the control. So this is going to be that delta density that we were talking about in the previous slides. I'm going to talk a little bit more right now, instead of going into too much more detail about the algorithm, about why it's so important to actually do this on single cell data, to do this on a single cell level for single cell data, because the sort of classic way that this has been done is instead of doing this per cell, calculate this sort of change in density or frequency of cells. By frequency, I just mean the number of cells in clusters of the data. And we're actually going to compare those two in one of the coding notebooks later today. It's a little bit fast on my computer too sometimes. So to exemplify why it's so important to do this on a single cell level, I'm going to introduce you to the data set that you're going to see in the last set of coding notebooks for today. These are 36,000 cells sequenced from zebrafish embryos that in the cordon condition, CHD, were injected with Cas9 plus guide RNAs targeting cordon, a gene required for developmental patterning. And then the control condition was exactly the same setup, but the cells were injected with guide RNAs targeting tyrosinase, which is required for pigmentation, but does not affect developmental organization of the embryo. We were talking about proper experimental design, and in this case, they actually did triplicates. So there were matched replicates. So cordon A is matched with tyrosinase A. They were sequenced at the same time. Cordon B with tyrosinase B, cordon C with tyrosinase C. And what we see here is that when we analyze this data set, there's actually no batch effect correction. What we see is that there's a huge amount of overlap between samples across conditions. So it's not like there's some cells that are present in one sample and then missing from another, but rather we have a sort of change in density across the data set that we'd like to be able to quantify. So in the published analysis of this paper, of this data set, what was done was the authors considered 27 clusters of the data that were used based off of a reference atlas, expert annotated, based off of gene expression. And the colors in this fate plot show the different clusters to which each cell was assigned. And we're sort of marking some of those populations here. And then what they did was actually just look at the fold change of enrichment. In cluster, tailbud/PSM, what is the number of cells from the treatment condition and the number of cells from the control condition, and then the ratio between them. So if, say, in that cluster, there were 100 control cells and 50 treatment cells, that ratio for that cluster would be a twofold increase. And this is a very discrete way to think about doing this. You're taking the graph, you're partitioning it into clusters, and then you're going and quantifying the difference within those clusters. But what we do is actually look at this on a single cell level. And so for every single cell, you get an estimate of the change in the likelihood that you would observe that cell in one condition or the other, in the treatment condition with respect to the control. And so what I want to emphasize here is that when we run MELD on this data set, the output is a ratio that goes between 0 and 1, where 1 means that you are 100% likely to observe that cell in the Corden condition with respect to the tyrosinase, or 0 means that you are 0% likely to observe that in the Corden condition relative to the tyrosinase. And what we can then do is look across replicates, for example, and start to say, so we actually don't do this just once for all of the cells from all the samples. As I mentioned before, we're estimating the density of each sample, and then we can start to look at how those samples are compared across replicates. And we're actually going to do this in much more detail in the coding notebooks today. And what you can see here is that we get this estimate of the relative likelihood for each replicate. And I want you to focus on two populations here, the notochord and the adaxial cells. And so because we calculate this on a per replicate basis, what you can see is that the notochord has relatively low values. They're blue across all of the replicates. So it's relatively depleted, regardless of which replicate you're looking at. Adaxial cells, which are muscle precursors, are actually enriched in one sample, not really affected in the second replicate, and then depleted in the third replicate. And so we would find that this is an inconsistent effect of the perturbation. And so we can actually look at not only the mean of these relative likelihoods across the data set, we can also look at the standard deviation of that measure of enrichment across every single cell. And for each cell, say sort of how confident we might be. We're not talking about statistical confidence. We don't actually use p-values because you don't have a really great null hypothesis or statistical model for how these likelihoods are getting generated. But we can at least get a measure of central tendency and of variation of the effect of the perturbation for every single cell. So I've been talking a lot about the fact that we're doing this for every cell and not just for each cluster. And the last thing I want to leave you with is why it's important to do that for every cell. This plot, you may not be familiar with the way that this is set up. We call this a jitter plot. It's a fast way to do something similar to a swarm plot or a violin plot. Each column here represents a cluster. These are those 27 clusters that were used in the published analysis. And the y-axis is the relative likelihood of observing a cell in the Corden condition relative to the tyrosinase. And each point here is a cell. And this gray is the average of those likelihoods for each cell. We're in the coding notebooks going to also look at the fold change measure, just the number of red to blue dots within each cluster. So if you look at this cluster OP, this stands for optic precursors. There's a full figure legend in the manuscript. But in this cluster, what you see is that there's relatively little variation around the mean for that cluster. So almost all of the optic precursor cells are depleted. But when we look across all of the clusters, we see that this is not actually the case for everyone. Some of these clusters actually have quite extreme variation in the amount of enrichment or depletion per cell in the data set. And so, for example, this tailbud presemitic mesoderm cluster not only has some of the most enriched cells in the data set on a per cell basis, but also some of the most depleted cells. And what's really important to note here is that this suggests that that cluster, although on average it's enriched, and we expect that. The presemitic mesoderm is known to be expanded. You actually visually see this tissue expanded in 24-hour embryos when you knock out cordon. So overall, it should be expanded, but actually there are some cells that are depleted. And when we go in and actually isolate just that cluster, we can do, generate a visualization of the data, and then see that in this region, there are some cells that are enriched and some cells that are depleted. And that there's not only heterogeneity in this fate plot, but a wide range of values that sort of is tied to that heterogeneity. And I'm not going to talk too much about this today because we're actually going to go much more into clustering next week. But if we go into clustering and then start to look at what are the subpopulations of this cluster, we actually can identify populations that we know are supposed to be enriched, and others like these adaxial cells that are supposed to be depleted across samples. There are actually experimental models showing that adaxial cells, which are embedded within the presemitic mesoderm, on a global transcriptional level, they're very similar cells exposed to very similar morphogen gradients, but they're distinct. They go on to become different kinds of tissues, that we see that different, subtly different populations of cells actually exhibit different responses to the perturbation. And so with that, I'm going to maybe wrap up with talking about MELD. This is actually a very long conversation. As I mentioned before, there is a link. I'll post on Slack a link to the full hour-long lecture if you're interested in hearing more about that. Right now, you can go and read preprint. So we just submitted proofs for this for Nature Biotechnology a week ago. There's a GitHub package online, and we're actually going to go through one of those notebooks today. And with that, I want to stop, take a little bit of a break. I'm going to introduce to you the coding notebooks that we're going to do and explain a little bit of why we chose the set of notebooks that we did, and really encourage everyone to go into the chat on Slack and please ask any kinds of questions that you have. We're going to start to break out into rooms now. People are welcome to stay in the main session if they'd like to ask specific questions. But for today's data session, what we're going to do, so we have 3 different kinds of notebooks. So the first one is clustering and denoising embryonic body data, which is broken up into two separate notebooks. So I am aware that we have not yet covered clustering as a topic in the lectures for this workshop. They should be relatively self-contained here, and you can actually see what we're going to be doing. If you have any questions, please don't hesitate to come out to the main session. We decided it's really interesting to use clusters as a way to sort of annotate cell types. We're going to just sort of assume that you understand a little bit of this idea that clusters sort of separate out parts of the data. We're going to get much more into how exactly you compare different clustering algorithms next week. But for now, we're just going to give you a couple that you can sort of just see how they work. But I think it's really important to understand how the data denoising aspects works on this data set. And you need to do some clustering, again, with this embryonic body data, I think, to get that out of it. We're also there's also one notebook here on doing batch correction in that PBMC data set that I showed you in the lectures. And then finally, there's a notebook on using MELD to do some kind of perturbation analysis. And I'll note as well that this notebook, it says a few times in there, I think, but this is a shortened version of a full tutorial that's available on the GitHub. And so I know that these are a lot of different kinds of topics, but there's only 3 notebooks for today. You'll see that we've actually got a lot for next week on Monday, which we'll kind of go through a little bit more. I think there's just, we find that there's a lot of content that people want to learn about in this course. And so it does mean that you maybe are a little bit closer to that kid with the fire hose in front of him than maybe the most comfortable, but I think it's the best way to learn all the content.